Good morning, everyone. It is a pleasure to be with you today on the Rare Disease Day in this webinar organized by the DREAMS project. We will be discussing advancements in clinical trial design tailored to the unique challenges of rare diseases. This webinar will feature contributions from researchers and practitioners related to the DREAMS project, but also involved in developing innovative methodologies. We will also have the opportunity today to hear from the perspective of a patient participating in clinical trials. Now I will tell you a bit more about the DREAMS project, which is a European project aiming to discover and validate novel shared therapies capable of treating multiple rare neuromuscular disorders for which there is currently no therapy. And this project is possible thanks to the collaboration of ISTEM, Quantify, Universidad de Coimbra, INSERM, Assistance Publique Hôpitaux de Paris, Technion, AFM-Téléthon, Zavala Innovation, and Samsara Therapeutics. After all the speakers are finished, we will start the Q&A session. So you can type all your questions in the chat anytime, but please mention who your question is addressed to, because this will help us get the best answer from the right speaker. So yeah, I can't wait to dive into the webinar. So without further ado, I hand over to Alexandre Mejat. And thank you all for attending. And Alexander, the floor is yours.
Okay, so thank you. I'm very happy to be the first introducing this webinar for DREAMS. And so without delay, could I have the slide with the agenda? So in the coming hour, you will have the chance to listen to 4 great speakers all around this question of clinical trials in neuromuscular diseases. First, we'll have a complete overview of the outcome measures in neuromuscular disorders clinical trials by the Professor Edoardo Malfatti. Then we'll learn about the lesson learned from a decade of Duchenne muscular dystrophy clinical trials from Gérald Perret. And we'll have also the very important testimony from 2 patients involved in the patient advisory board of the DREAM consortium to have their perspective on their role and participation in the building of a clinical trial protocol. And just before, I will quickly, in just 3 slides, try to tell you why there is a need for innovation in clinical trial methodologies for muscular disorders, but more generally in rare diseases. Could I have the next slide? So my name is Alexandre Mejat. I'm working for AFM-Téléthon, and I'm one of the partners of the DREAM consortium. And with Wael Kazen from AFM-Téléthon, we are in charge of managing the work package 6, which aims to design an innovative clinical trial for neuromuscular disorders and rare diseases in general. And as I said, I will try to tell you why there is a need for such innovation. Next slide. So on this slide, I just showed you what is a classical, very classical way of doing clinical trials. So usually you identify a potential cure or treatment in a preclinical step where you test it on different models, such as cell lines or animal models, and then you want to test it to humans. If it's a brand new drug, you don't know what could be the effect of this drug, even in healthy people. So usually clinical trials start by what we call a phase 1, which is a phase where you will involve only a few patients. You see on this slide, they say less than 10. It's not necessarily people affected by the disease you want to treat. It can be healthy people. And you just want to check that at least your treatment will not create adverse effects. So will have no negative impact on people. If you see no negative impact, then you will move to phase 2, where you will start looking for efficiency or efficacy of your treatment and also evaluate the safety of your treatment. You will usually do it on a bigger group of people, between 20 and 50 participants. and which will, of course, include this time people affected by the disease, as you want to evaluate the efficacy. And to also measure this efficacy, you usually divide your group of people in two groups, one receiving the treatment and one not receiving the treatment, but receiving what we call a placebo, which is a known drug to have a reference. And if in this phase 2 you can observe a great safety and efficacy, you usually move to phase 3, which includes even more patients. And here you see it includes until 200 participants to completely confirm the benefit and safety of your treatment. And it's only after this phase 3 that you can usually ask for this drug to become a medicine available for patients. And after this treatment is available for the whole community of patients, you usually can have a phase 4 where you will have a follow-up on all people treated by your treatment. So this is a classical way, phase 1, phase 2, phase 3, with an increasing number of patients involved in the clinical trial. Could I have the next slide, please? So why do we need to do it differently in neuromuscular disorders and in rare diseases? First, as you saw in this first slide, this kind of clinical trial requires hundreds of participants. And unfortunately, neuromuscular disorders are part of what we call rare diseases, which means that there are very few patients affected by this disease. And the classical way of doing clinical trials involving hundreds of participants cannot be applied to rare diseases. So we need to do it differently. Click, please. You also saw that there are several phases, phase 1, phase 2, phase 3, each of them taking a few years. So when you accumulate all these years to go through the whole process is a very, very long process. And as you know, our pathologies are quickly evolving. And so we need to go as fast as possible to provide treatments to patients. And so we need to find a way to reduce this time and not be blocked by these phase 1, 2, and 3. Click. As it's defined with very clear phases, it's usually very complicated to modify what you define in each phase with what you define and submit it to regulatory agencies. And so if there's anything during your phase which goes wrong, or if you have new scientific data or new drugs that you would like to test, you theoretically cannot modify your current clinical trial and need to start a brand new one, which again takes a longer time to be implemented. Next click. And so finally, when you accumulate the number of participants, the time it takes, the number of phases, it can become extremely costly, which can be a clear limitation to simply start the clinical trial if you cannot find all the funding necessary to do the clinical trial. So what do we try to do in DREAMS to solve this issue? Next slide. So we try to combine innovations in a clinical trial protocol, which will be adapted to the DREAMS consortium, but more generally, which could be also adapted to rare disease in general. Click. So first, we will start with 5 rare neuromuscular diseases. So we will not look for a treatment for only one pathology, but for 5 different pathologies. To do so, we will collect cells from patients and test drugs on these cells to hopefully find drugs which could have a positive effect on the cells of these patients affected by these 5 rare neuromuscular diseases. Click. And hopefully we'll find at least one drug which can be efficient for more than one rare neuromuscular diseases. In the perfect situation, we would find one treatment which could have a positive effect on the 5 rare neuromuscular diseases we started from. Click. And once we have this drug, we will also use artificial intelligence to try to find other drugs which could have the same benefit on the 5 rare neuromuscular diseases. Click. And knowing the drug we found, we can also look for similar drugs which could have also a positive effect. So by this process, we hopefully will have one or more treatment which will be usable for one or more rare neuromuscular diseases. Click. And so now how can we innovate? We will try to test one treatment for several pathologies. In our case, at least 5 rare neuromuscular diseases. When we test one drug for different pathologies at the same time, it is called a basket trial, and that's a way to optimize your clinical trial. Click. As we will start from 5 rare neuromuscular diseases, we will also apply very specific mathematical strategies to make sure that even with a small number of patients tested, we will have very robust statistical results. Click. The kind of drug we will test are not brand new drugs. They are what we call repurposable drug. A repurposable molecule is a molecule which has been already developed for another pathology, which means that all the information needed to do what we call a phase 1 are already done. So we can hopefully directly start our clinical trial in phase 2 and propose a clinical trial which will not be blocked by phase 2 and phase 3 in different and separate time, but go from phase 2 to phase 3 in a very fluent way. And that's what is called a seamless design, where you go from phase 2 to phase 3 with no stop. Click. And finally, as we will use artificial intelligence to find other drugs which could have the same effect or other pathologies which could benefit from the same drug, we may have to integrate these new drugs at some point in our protocol. And so we need to have a protocol which can adapt to new discovery done during the time of the project. This is called adaptive design. And so our goal in the DREAMS consortium is to build this kind of innovation in a protocol adapted to the 5 rare diseases in the consortium. And so now one of the challenges to build this kind of clinical trial is to find what we call outcome measures. So a way to measure the efficiency of the treatment. And so to talk more about these outcome measures, next slide, please. I would like to introduce Professor Edoardo Malfatti. So Professor Malfatti is a full professor on neuromuscular disorders and histopathology at the APHP Henri Mondor Hospital. He's also involved in the international networks because he's the chair of the EuroNMD Neuromuscular Pathology, a specialized group. He has more than 50 years of experience in different countries in the world, Italy, France, and Sweden. And he has been discovering genes responsible for different neuromuscular disorders. And since these discoveries, trying to understand more carefully the mechanism of these different pathologies. And so now he will tell us more about all the outcome measures that can be used in neuromuscular disorders clinical trials. Edoardo, the floor is yours.
Do you see my presentation? It's okay?
Yes.
Thank you very much. So today I will make an overview of the outcome measured in neuromuscular disorder for clinical trials. And I decided to organize this presentation making a short introduction of neuromuscular disorder. Briefly describe what is an outcome measure and a clinical trial, although Alexandre already explained in detail how clinical trials are designed. I will talk a little bit about the general outcome measure that could be applied and have been applied to different neuromuscular disorders. I will define a little bit biomarkers and imaging techniques that can be used to assess if a drug is effective in a patient with neuromuscular disorder. And then I will describe 3 different pathologies where there are already approved drugs and how the outcome measure have been selected in order to demonstrate the effectiveness of this drug in order to go into conclusion. So as you may know, a neuromuscular disorder, a neurologic disorder that affect the different segments of the peripheral nervous system, either the motor neuron, they can affect the peripheral nerve, the neuromuscular junction of the muscle fiber. These are very heterogeneous disease, but they are presenting with a common symptoms, that is the muscular weakness. Muscle weakness provoke a functional decline that go directly impacting the patient independence and daily activities, leading in extreme case to the loss of ambulation with the need to a wheelchair to move. Neuromuscular disorder, as stressed by Alexandre, rare or ultra-rare disease, but these disorders taken all together are more common than what we think, as there are 8,000 different rare or ultra-rare disease that affect worldwide 350 person, and only in Europe there are around 30 million of people affected by rare or ultra-rare disorder. And 50% of this disease affect patients from birth and during all their life. And of course, our final goal is to find a therapy for all neuromuscular disorders that are rare disease, with some limitation in the development of a clinical trial, as already told by Alexandre. I don't need to go deeply in this, into these slides, but with a clinical trial, what we want to do is to evaluate if a new treatment is safe, if can have an effect, if we have elements that tell us that we have to vigilate some pharmacological eminence. And these are new intervention in humans that need to be set according different phase of development. To do so, we need the outcome measure. And what is an outcome measure? So there are standardized tests that are used to assess various aspects of patient health and can give us responses in order to see in a clinical trial if a drug is effective or not. The outcome measures are essential for a clinical trial because they help us to gain a standardization, so to enable a comparison of results across studies and different patient groups. And in rare disorder, with this basket concept, is very important to be sure that we are observing the same effect in a limited group of patients. They give us information about the treatment efficacy, measuring in a precise manner clinical changes in responses to therapy. And then it helps also us to monitor the disease of progression and help understanding the course of condition over time. Are fundamental for trial design optimization because they determine the required sample size and the study duration, and then finally help us in the therapy validation, supporting the approval of treatment for clinical use by the different agencies like FDA or EMA in Europe. Ideally, these outcome measures should be non-invasive, should be quantitative, cost-effective, and simple to perform in order to allow reproducibility. We have a large heterogeneity of the outcome measure that have been used till now, and new elements are coming thanks to novel technology. We can distinguish between a generic outcome measure that can be applied to multiple disorders. We can have what we call the biomarkers and the imaging outcome measure, where the biomarkers are taken by invasive procedure like a blood sampling or, for example, a muscle biopsy. And then we have a disease-specific outcome measure that are tailored to specific neuromuscular disease. And I will give you example of 3 disorders where we use different outcome measures in order to assess the progression of disease and the effectiveness of the drug. And then what very importantly, we have the patient's reported outcome measure. In many neuromuscular disorder, we have a burden with a limitation of the quality of life, with fatigue that is very important, for example, in myasthenia gravis, and we have an alteration of daily functioning. Considering all these elements with a specific scale, with specific tests, is pivotal in order to select the best outcome measure. Quality of life in genetic neuromuscular disorder have been studied and tested using this test, the quality of life neuromuscular disorder. This is a scale that has been designed specifically for patients with slow progressive neuromuscular disease with genetic predominant muscular damage. It has been developed through a rash operational validating measure, and it involves three key domains: the impact of the disease and the muscular weakness on physical symptom, what is the self-perception of the disease, of the handicap that is caused by this disease, and what are the consequences on the activities of the daily life and the social participation. This scale has some limitation because they need a psychometric validation, so it's not, the data should be interpreted under this light. There is another scale that has been developed for neuromuscular disorder that is called active limb activity limitation. This has been developed and validated in patients with neuromuscular disorder. It is reliable and reproducible with a good sensitivity to change, and is useful in also in clinical trial and research setting to characterize how the disease progresses and how medical and treatment can limit this progression. It is built using 22 items that assess the daily activities, with 4 questions specific to children and 4 specific to adults. We have 4 different possibility of response: impossible, difficult, easy, and a question mark if this activity is not attempted in the last 3 months. The limitation of the scale is that can measure different magnitude of clinical changes across various neuromuscular diagnoses, and specific questionnaires may be recommended for the clinical trial targeting specific diseases. But let's see, regarding the muscular weakness, what is a simple, are 2 simple tests that measure the walking abilities of the patient. The most widespread tests are the 6-minute walking test. This is a validated measure of exercise capacity and motor function. Has been widely used in several neuromuscular disorders where the patient are ambulant, and it measures the maximum distance a person can walk 6 minutes over a 25-meter linear course. It's pretty simple to perform, but of course you need a train by the physical therapist or the research assistant in order to be sure that it's done in a timely way. The other test that is shorter, and for example, it's more easy to make performed by younger population. Of course, children that need to walk for 6 minutes can be easily tired and lose their attention, while 10 meters can be easier to perform. And it assesses the walking or gait speed in meters per second over a short distance. It can be employed to determine functional mobility, gait, and also vestibular function. Another a set of elements that we can test and you can use as an outcome measure are the tests that measure the balance and the postural transition. Some examples are the time up and go. So we ask the patient to go up and go, and this can assess the balance, the gait speed, and the postural transition. The participants stand up from a chair, walk 3 meters, turn around, walk back, and sit down. And there is a cutoff of 13.5 seconds that suggests an increased risk of falls in older adults. We can have measure like the 30-second sit to stand. These evaluate lower limb strength by counting how many times a person can rise from a chair in 30 seconds. We can use a simple set of stairs, usually 4, 10, or 13, or 12, sorry, that can measure the time to ascend or descend. And this is important to test complex motor function. in particular the lower limb power, strength, and functional mobility. We can have time stair climb test, similar to the previous test, but with a focus on the speed while ensure the safety of the patient. Can be done with an handrail support on without, depending on the patient's ability, and this also can evaluate the risk of fall, the functional independence. And then we have more complex form of measure the balance and postural transition with the platform of posturography. The strength can be tested both manually. We have a simple manual muscle test with the MRC scale that tests the muscle strength in different group of muscle, the proximal, the distal, but of course these can be operator dependent. And then we have different ways of utilizing dynamometers that can test the strength. For example, these isokinetic dynamometers that are advanced electromechanical machines that are controlled by microcomputers. They measure torque output of a muscle group during movement at constant angular velocity, and they are equipped with force and position sensor. We have a grip test and hand dynamometers that you can see here. These measure the maximum voluntary isometric static force produced by specific muscle groups during contraction and contact angular position. And these are very handy and can be used during in a short time lapse during a clinical trial. But let's talk a little bit about the biomarkers. We define a biomarker as a molecular, cellular, or biochemical change that can be accurately and repeatedly measured, and it monitors a physiological process, pathogenic process, pathologic process responses to pharmacological intervention. The classical example is the serum creatine kinases, which is a marker of degeneration of the myofibers. These enzymes are present in different tissues and organs. What we are testing specifically for skeletal muscle are the CK-MM, the myo form, and we have cutoff values that have been calculated based on the sex of the patient and also based on the ethnic origin, as we know that the African population has normally higher level or serum CK. And what is important that these tests can be used especially in muscular dystrophy and in clinical trials for example, Duchenne muscular dystrophy, serum CK level reduction or normalization is an element that we evaluate. Regarding the imaging technique, the muscle MRI imaging have been used widespreadly to quantify the lesion that consists in fibrofatty muscle degeneration. Here I put an image of the progression of the fibrofatty tissue degeneration that is seen as a whitish area inside the muscle in patients with Duchenne muscular dystrophy. And what we can do, applying specific techniques that are called Dixon, where we calculate, subtracting the water signal, the percentage of fat, how this evolution in a clinical trial. There are also more invasive markers. One that can be used is based on the analysis on the muscle biopsy with techniques of immunohistochemistry, the staining here, for example, the dystrophy. You can see that in a Duchenne muscular dystrophy patient, you don't have this marker. The reappearance of these markers after a gene therapy, for example, is a marker of effect at tisular level. But let's see a little bit some example of disease where there are approved drugs and how the different scale and the different outcome measures have been used to demonstrate the effectiveness effectiveness and the introduction, allowed the introduction of these drugs in the clinical practice. The most successful example of disease in the field of neuromuscular disorder is spinal muscular atrophy. Is a motor neuron disease with different form of disease that provoke different degrees of handicap, with the severest form type 0, where the patient presents a prenatal form. Then we have type 1, where the patient cannot walk if they are not treated, to milder form, the type 4, for example, where patient develops symptoms in adult. What we have now that therapies arrived at the bench of the patient with 3 approved therapies. Splice affecting, intervening at the level of splicing of SMN protein that is defective in this patient, or through gene therapy that replace the gene that is defective. And here in this part, I put different scales that have been used to assess the effectiveness of this drug and that allow them to arrive to the patient. Some example, the Hammersmith Functional Motor Scale, different protocol, expanded to, for example, ambulant patient for the HFMCE, or the revised Hammersmith scale. What are the ability of this scale? The scale can capture motor function chain for very weak SMART2 patient to patient type 3 that are ambulance, and even changes in very severe patient can be detected. It is structured in 69 items where we ask the patient to perform a specific movement, and they have a 3-point like a scale. And the materials that we need to perform this scale are standard stairs, step, we have 230 centimeter separated lines and a 10 meters line to test the ambulation. What are the limitations of the scale? Of course, we need to evaluate a different time points and to evaluate the effectiveness of the drugs in changing this parameter for at least 6 to 12 months to be sure that there is a clinical meaningful modification. Another scale that has been developed here in France is the motor function measure, that is MFM. It exists in 2 forms, the 32 and the 20. This is a quantitative scale, very globally assessing the motor function in individuals with neuromuscular disorder. It is available in many languages and it has a pediatric version. What we are testing with this scale, we are testing 3 different dimensions. Dimension 1 that correspond to standing, position, and transfer. Dimension 2 that allow to test axial and proximal limb function, and dimension 3 that is focused on distal motor function. The score is done in 4 points. It requires simple tools, for example, a tennis ball, coins, a timer, and the limitation is that there is a possible ceiling effect where we do not capture, for example, the limited change in distal weakness on this patient. And to specify and to really investigate the alteration on the upper limbs strength in patient with SMA, there is this scale that is the revised upper limb module that has been developed to assess clinically meaningful point determined as an amelioration and an upgrade of the scale of 2 points. It is structured in 37 items with 3 points for every item, and it needs a simple material. And this scale has been used very widely in order to determine the disease duration impact and the modification after therapy. Let's talk a little bit about Duchenne muscular dystrophy. I don't need to go into detail, telling also only that this is a genetic disease linked to a deficiency of this protein, that is dystrophin. It is X-linked, and it starts before the 4 years, and without an intervention. Now there are steroids that are approved for this drug, and 2 different drugs, Talorem with exon skipping, and nonsense read-through, and drugs for exon skipping in United States, but there are no effective drugs to cure this disease. Specific scale for the Duchenne muscular dystrophy have been elaborated, specifically the North Star Ambulatory Assessment, where we detect in clinical trial the changes due to increased steroid dosage. The structure is also 17 item with 3 points like a scale, and the material required is 15 centimeter step, 10 meters line, and a stop short. The limitation was that was used in trial comparing treated versus untreated patient. Careful matching of baseline of age is necessary in order to assess the difference. And then now we have a long natural history, reference natural history, but using control groups was necessary in the first phase of development of the scale. According to the novel technologies, I would like to mention the stride velocity at 95th centile. This is a digital measure of maximal walking speed that is captured by this wearable device, and it represents the fastest 5% of the strides that are taken over a given period. That is the last part of the stride movement. This is very important because it's objective. It's a real-world assessment of ambulation without the need of a scale that is operator-dependent, and provides sensitive detection of functional change over the time. Potentially complements traditional scale, for example, the 6-minute walk test or the 10 minutes. And the objective of the scale is the continuum, that is to allow an objective, a continuous measure of ambulation, capturing real-life working ability beyond traditional time tests. The impact on clinical trial was to provide quantitative and reproducible mobility assessment, help to validate treatment efficacy, support regulatory decision-making new therapies. Just a mention of the performance upper limb 2.0. This was originally developed for individuals with dystrophinopathies, but can be applied to other myopathies, like, for example, in Limb-Girdle muscular dystrophy. In this scale, we have 42 or 22 items with a different set of weights, 6 token, pencil, and plastic cup. And then you need to have sheet papers, plastic container, push button light, and it's very focused on the upper limb function, and this is particularly interesting for patients in wheelchair. The limitations are some ceiling effects that affect the measurements of stronger individuals, and a top score on the entry item may negate the need to complete the rest of the scale. But let's talk to a completely different pathology, that is myasthenia gravis. In this disorder, we have a great fatigability in patient, and we have an alteration of the ocular, bulbar, and proximal and respiratory muscle. This patient usually retain ambulation, but we can have a form that lead to very severe fatigability. In this disorder, we have recently got new therapies in complement of the anticholinesterasic, the immunosuppressive therapies, and these drugs have been tested thanks to different scales. The scales that we usually use, and that is a myasthenic muscle score, it consists on a 100-point scale with 20-point improvement that define the treatment response during the clinical trial. And you can see here the French version, where we ask the patient to perform a resistance exercise for the upper limbs, for the lower limbs, and then we deeply evaluate the presence of ocular involvement, like for example ptosis or diplopia, or the bulbar involvement with swallowing alteration, chewing alteration, or speech alteration. The scale can be operator dependent and has been really complemented by the quantitative myasthenia gravis that is commonly used all around the world. As you can see, we have a set of evaluation that are pretty detailed. The advantage is that almost all objective measures of fatigability are applied. The items are dedicated to distal upper limb strength and the bulbar sector. The cons are that there is a long assessment required by clinician trained to use specific equipment, because this scale also includes the spirometry of the patient. Another scale wants to quantify the quality of life in myasthenia gravis patient. This scale is called MGQOL 15 and include 15 questions related to how quality of life is affected by myasthenia gravis. And these involve three domains: the physical domain, so the limitation for physical activity; psychological, patients that, for example, have important dysphagia are limited in their social activity, like, for example, going out for dinner with family or friends; and the social impact that can be very high. And also there are questions about the perception of the disease, like, for example, the level of frustration provoked by disease related to the different symptoms. And with this was my last slide. In conclusion, as you can see here, we need for future clinical trial to identify a reliable and disease-specific outcome measure that are able to capture change determined by a new drug. We need a patient compliance. I show you that this test can be kind of time-consuming, burdensome for the patient, because we need to ask them to repeat some tasks and to perform long tests. It is important to complement and to add as outcome measure the patient reported outcomes that provide a crucial insight into the real-life functional impact. Of course, we want to cure a disease, but we want our patient to be functional and to be fulfilled in their life. And I show you an example of how new technology with the stride velocity, with the wearable device, can offer new objective outcome measure for improved monitoring. And with this, I conclude. I would like to thank all the members of our team and all the funders of our different research projects. Thank you very much.
Thank you, Edoardo. We see how diverse of these tools to measure the different efficiency of the treatment and how challenging it can be. And so we will now move to a concrete example of clinical trials for Duchenne with a talk from Dr. Gérald Perret. So Gérald is a program and clinical development director at Genethon, one of the laboratories funded by AFM-Téléthon to develop gene therapy. He has more than 20 years of experience in the pharmaceutical industry. He has been working for groups like LFB Groups or Sanofi. And he has both a PhD in molecular biology and a master degree in economics. And he will now tell us more about the lessons learned from a decade of Duchenne muscular dystrophy clinical trials. Gérald, the floor is yours.
Okay, thank you, Alexandre. I'm just checking if you can see my screen.
Yes.
Okay, perfect. Thank you. So here's the agenda with a brief introduction on the disease, followed by, let's say, the 10 chosen lessons from 10 past years. So the Duchenne muscular dystrophy is X-linked recessive neuromuscular disease caused by a mutation in the gene encoding for the dystrophin protein, which is a major component of the dystrophin-associated protein complex that you can see here. And because of this mutation and the related loss of function, in Duchenne patients, the muscle fiber regeneration becomes impaired, leading to deterioration of muscle integrity, resulting as the first onset of the symptom around 4 years of age, in first difficulties with mobility, including the loss of ambulation around 12, thoracic scoliosis, need for respiratory assistance by 12, and eventually the death by respiratory failure or cardiac failure between 20 and 40. So it's definitely a devastating disease. With respect to this devastating disease, numerous of clinical trials have been launched, especially during the last 10 years. And these 10 years have taught us many lessons. And I propose to focus on what I consider to be the 10 most important, which are listed here and which I will elaborate on. Starting with the heterogeneity. Indeed, DMD is a complex and heterogeneous disease with significant variability in disease progression, as illustrated here, with a graphic from Francesco Montoni's work that represents the main 4 categories of disease trajectory monitored with a scale that I will discuss later. And this heterogeneity is particularly challenging for the youngest patients or those in the late ambulatory stage. For the youngest, the situation is probably the most complicated, with, as you can see, opposite trajectory. Maybe if I focus, I have an animation, yes. With different trajectory, opposite, I would say opposite trajectory, with some still improving and others decreasing. And you can imagine that for such a population, the challenge of an external control is high. If, for example, by lack of chance, this control is composed mainly of an improving population, that will, at the end of the day, hide any effect of your treatment. Or, on the contrary, if the population is composed mainly of decreasing profile, in this case, that may overestimate the effect of the evaluated drug. This kind of situation was suggested as a limitation encountered in several products which eventually failed in phase 3 randomized placebo-controlled studies. And this challenge could be overcome by very rigorous propensity-based matching of untreated patients, including, for instance, the class parameter, if already identified, or with the same logic to have, I would say, inclusion criteria based on disease trajectories. In addition to this parameter regarding the trajectories of the disease, there is something else that has to be managed, which is a consistent standard of care, especially in terms of dose and type of corticosteroid, and that is probably another key to limiting this complicated heterogeneity. With respect to biomarker, relevant biomarkers are obviously crucial for monitoring disease progression and treatment efficacy. The identification and use of factors which are strongly predictive of disease progression is imperative to support clinical and allow conditional and accelerated approval, but it's not easy. Edoardo mentioned the microdystrophin expression. Indeed, microdystrophin or dystrophin expression could be considered as very relevant surrogate endpoints, but there are limitations, including the lack of representativity for the whole muscle tissue because it's only a small piece of muscle, the lack of standardization across all the methods that are using to measure the positive or the quantity, the positive fiber or the quantity of microdystrophin or dystrophin expression. And the last but not the least, taking of the burden that represent biopsy for Duchenne patient, it's not something that could be repeated. Maybe serum biomarkers such as creatine phosphate kinase be more relevant to assess globally the impact on muscle, but it's no, the variability of this biomarker is high, and considering the progression of the disease is critical because an advanced form is an advancer of the disease irritated by a natural decrease of the CPK. Maybe as the last example of biomarker improvement across clinical trial, the advances in mathematical modeling applied to medical imaging have provided some elegant solution. In particularly, I would like to highlight the MRI, the quantitative MRI, to assess fat fraction that is performed without any significant patient burden. And as for clinical outcomes that I will just discuss after in the next slide, it's clear that complementary and multiple biomarkers may offer some solution to have, I would say, a true vision of progression of the disease and so impact of the therapeutic used in clinical trial. So speaking of outcomes, this field has considerably changed the last 10 years since the development of the performance of upper limb, the PUL. The first, I would say, obvious consideration is to choose outcomes with respect to the stage of the disease. The stage of the disease you are considering is a trial. Loss of the ambulation, initiation of ventilatory support, or loss of hand to mouth. It's clear that so far, in the last 5 years, I would say, we observed regarding the drug development, a focus on late ambulatory phase. And in this context, the NSAA, North Star Ambulatory Assessment, appeared as a gold standard. And you have a description here of this outcome in this table. It's a 17 item clinical rated tools that measure ambulatory performance. Each item can be scored as 0, 1, or 2 with the best performance. And the total score of the NSA range from 0 to 34. So with the highest score at 34. And for the record, there is an equivalent ambulatory assessment for the LGMD called NSAD. In addition to that, individual time tests have been widely used, as well as in the clinical trial conducted in ambulatory or late ambulatory population, as for example, the 10-meter walk or run, the time from 4, and major point. Oh, sorry for that. It's not. Yes, a major point to consider for this. I'm sorry, it's not the. In this slide, sorry. So an important point as well is the confidence of regulatory agency in these outcomes. And to ensure that the use of outcomes that have been qualified by FDA, EMA, or both is definitely key. As it is for one of the most recent outcomes, like the stride velocity or SV95C, that is based on the use of a device that the Duchenne patient are bringing during a couple of days. at different times during the trial to register real-life motions. No less importantly for this outcome, a robust and standardized definition of what is a minimal clinically important difference. And maybe more precisely, what this MCID, minimal clinically important difference, is related to a time frame, because the difference observed in comparison with external control in 1 year is very different to what could be observed after several years, the same level after several years. Regarding new generation of outcome, we can mention as well the use of video. For instance, the Duchenne video assessment, DVA, which is a new video assessment outcome which is recorded by caregivers for boys with DMD, and the DVA captures the ease of movement and the acquisition of compensatory actions across different curves. And every video is sent for a quality review by data monitor and scored by DVA certified physical therapist. We can mention as well the ability captured through interactive video evaluation, active game. And as I said, for the biomarker, there is for outcome as well a rationale to use complementarity, multiple endpoints. But in this case, in addition to the burden for patients, there are concerns that regulatory agency may advise clinical trials to meet the full statistical power for all. So that would mean that in other words, to have primary and secondary endpoint being treated equally. So asking for statistical significant beyond positive trend, and thereby increasing the chance of failed trial in potential effective drug, because you need to have the statistical power over all the clinical endpoint, and that could be quite difficult. So I would say that the last clinical trial have shown the critical importance of duration for outcomes measurement and long-term follow-up. If we take the example of glucocorticoids, which have both an anti-inflammatory effect and an anabolic effect in the dystrophy muscle, this result is in improved muscle strength and function, which can be measured within weeks or months of initiation, but definitely does not prevent the long-term progressive muscle damage characteristic of the Duchenne muscular dystrophy, leading to limited long-term impact on the disease progression. Given that, an outcome measurement during only 6 months is probably unacceptable. Longer for outcome is probably the best, but that raises issues in cost, feasibility, and burden for patients. In this context, definitely compromise should be made. Within the concept of treatment meaningfulness that I just mentioned, there is as well the timeline of effect, short versus long-term effect, and how this interacts with the age of treatment initiation, which is needed for effective treatment. And we can consider in further clinical trials the concept of, I would say, best treatment time approach for both methodological approach and labeling consideration. During the past 5 years, we observed a rise in clinical trial using a similar approach of microdystrophin gene therapy. This concept is based on the challenge of the size of the dystrophin gene, which cannot fit within the capacity of gene vector. Today, there are 4 clinical trials ongoing based on this concept of microdystrophin, with difference in vector, promoters, and construct, as you can see on the right illustration, the right picture. And in this clinical trial, we observed immunolaterative adverse event immune-related adverse events, and most of them were, I would say, quite unexpected, like the complement activation or immune reaction against the transient. And it was really the opportunity to identify a new way of management, of monitoring this kind of adverse event. And I'm convinced that today these adverse events are, I would say, well controlled and could be likely applied to this new control and management could be likely applied to other gene therapy in neuromuscular disease. Gene therapy brings some spectral results, as I'm going to share, as in this patient in this video from a clinical trial sponsored by Genethon. It's a patient that had been treated 2 years ago when the first onset of the disease appeared. And this patient reached the maximum of the NSAIDs that I just mentioned in the outcome slide. And 2 years after the treatment, and still at the maximum. Obviously, despite this kind of very promising result, there is today still an open question regarding the durability of gene expression. And for that, the future will bring answers. Regarding the antisense oligonucleotide, even for the approved one, there is definitely improvement required due to their low efficacy and various methods, and there is various methods to improve that. Administered antisense oligonucleotides circulate in the bloodstream and are distributed through the body. During this process, the efficiency of ASO delivery is become, I would say, a critical factor in their effectiveness. And to enhance this delivery efficiency, over all the tissue, there is some conjugation that have been developed to increase, for instance, lipid affinity, to improve membrane permeability, and allow an absorption from the gastrointestinal tract, in addition to chemical modification to prevent the endosomal sequestration, and everything is ongoing investigation. We can give, for example, the use of locked nucleic acid, Eleonora, to enhance the binding over the genome. Considering the result and design of several disease-modifying approaches, I truly believe that at least the corticosteroid will be maintained as a life treatment. And this observation supports the interest of future combination approaches that make perfectly sense if you consider the different components of the Duchenne muscular dystrophy pathophysiologies, which are structural, fibrosis, inflammation, and impact on signaling. And consider the different components that make perfectly sense to have in the future clinical trials that will use a combination of drug under investigation. For example, a combination of gene therapy or exon skipping with HDAC inhibitor, with corticosteroid, including last generation of corticosteroid, and potentially in addition to a light immunosuppressive regimen. That could be a very complicated clinical trial, but with maybe some key outcomes. Regarding the flexibility of regulatory agency, it's clear that today most of these flexibilities have been observed on FDA side, including accelerated approval pathway, a strong consideration of surrogate endpoints, and maybe a last example that I can mention is the primary endpoint that have been negotiated between Regenix Bio and FDA, which is based on a level of microdystrophin expression that has to be only over 10% as a surrogate endpoint. In addition to this, I would say, regulatory flexibilities, the PRV is really an incentive for a drug developer. The PRV, Priority Review Voucher, which is granted for the approval of a drug for an orphan and pediatric disease, provides significant incentive as it could be valorized around 100 million euros. On the European side, there is true that we observe an increasing interest in the use of robust surrogate endpoints for both market application and HTA evaluations. Regarding the safety management, I mentioned what we observe with gene therapy regarding the immune-related adverse event, and this kind of specificity around related to Duchenne muscular dystrophy clinical trial may be explained by two reasons. First, we are trying to target large and sensitive tissue. Large because we are talking about all the skeletal muscle. Sensitive, we are talking about cardiac and respiratory muscle. And the Duchenne muscular dystrophy is characterized by an inflammatory background that could support specific and non-specific immune reaction, as the one we observed during the clinical trial using gene therapy. So definitely, the lesson learned from a clinical trial include significant improvement in exclusion criteria, management and monitoring of serious adverse event, and some significant work with respect to the prediction of this kind of events. One of the last questions I propose is it is the last actually, and the last but not the least is the importance of collaboration in clinical trial. One of important lesson in Duchenne clinical trial that I would like to share is a collaborative approach between competitors involved in gene therapy field in order to better understand recruitment, in order to better understand what has been, I would say, involved in the immune-related adverse events that I just mentioned. And this example that lead to a publication here, you can see here, has been really appreciated by regulatory agencies that strongly encourage to have similar position across competitors, including approach for safety management and monitoring, surrogate biomarkers, standardized clinical outcomes. And that is something on which we are strong expectation, sorry, from the regulatory agency. If the support of patient organization regarding the choice, the evolution of the next generation of outcome, as well as the evaluation of the burden of the clinical trial is definitely key. I consider that more could be done. For instance, in order to optimize the recruitment, it would be useful to communicate to all advocacy groups the procedure that are followed by the clinician to recruit patients in competing trials. That could definitely improve and rationalize the recruitment for patients. Oh, sorry. So, in conclusion, we can definitely say that the past 10 years have been the opportunity to numerous lessons for the next clinical trial, including considerable improvement in the understanding of the variability of the disease. We discussed that the importance as well to have a standardized and I would say confident definition of minimal clinically important difference for clinical outcomes. And for this outcome, there is still avenue, definitely today, for new outcomes, especially with respect for the clinical trials that will address non-ambulatory patients. There is some rationale of using multiple endpoints or multiple biomarkers, despite the limitation of burden for clinical trials that have to be considered. I mentioned as well the consideration that you need to have with respect to the duration of the clinical trial and the importance of the long-term follow-up study after the first part of the clinical trial. We discuss as well about the flexibility of regulatory agencies that recognize the unmet medical need in Duchenne muscular dystrophy, despite the fact that today most of these flexibilities are observed in the US. And we discuss as well the impact of collaborative approach between sponsor, academia, and patient organization. And as I discussed, there is already today some great hope, I would say, with the first promising result with disease-modifying drug as gene therapy and potentially as well exon skipping. And it is my last slide.
Thank you very much, Gérald. It was a very complete and extensive presentation. And we see we can learn from what has been done for pathologies like Duchenne, but also that there are challenges. And one of the things you mentioned was the important collaboration, not only with between clinicians, researcher, or pharmaceutical company, but also with patient organization. And in DREAMS, we wanted absolutely to have the patient perspective taken into account, more particularly as we are dealing with 5 different diseases with different impact in everyday life. So I'm very happy to know welcome 2 of the members of our patient advisory board, Eleonora Cugudda and Orla Lynch. And first I will invite Eleonora Cugudda to share with her perspective. I can say Dr. Eleonora Cugudda. She's from Italy. She's a distinguished researcher and clinician specializing in public health and neuromuscular, and she's also a neuromuscular patient. She has published numerous scholarly articles and research papers in reputable medical journals. But being a neuromuscular patient, her medical career has been also enriched by her personal experience. And this dual perspective enables her to approach medical challenges with a comprehensive and insightful view. She's very actively involved in various international collaboration and research initiatives aimed at improving the lives of individuals affected by neuromuscular disorders, and more particularly by Emery-Dreifuss muscular dystrophy. So I'm very happy to leave the floor to Eleonora.
Thank you very much, Alexandre. It's a pleasure to be part of this meeting on Rare Disease Day. I am a medical doctor, as you said. I'm specializing in public health, currently working in medical management at a hospital in Turin, Italy. I have Emery-Dreifuss muscular dystrophy, which causes significant muscular weakness and cardiological issues that I face in my everyday life. As you said, in addition to my medical work, I lead a national community and an international community of patients, and I'm very, very happy to be here right now. I joined the Patient Advisory Board of DREAMS because I strongly believe that involving patients in research benefits everyone. It brings a human face to scientific work and offers hope to those directly impacted by these diseases. Being part of this committee allows me to contribute in shaping research in a sort of way that truly reflects the needs and experiences of patients, ensuring that the voices are heard and that the progress is made with them at the center of it all. Within the Patient Advisory Board and the DREAMS project in general, I hope to contribute with both my expertise as a doctor and as a patient, providing a comprehensive and insightful perspective. My expectations from the DREAMS project are to take even a small step, finding new pathways to therapy for neuromuscular diseases. In this webinar, we've heard about the impact of pharmaceutical therapies, especially for Duchenne muscular dystrophy, that has been changed. Pharmaceutical therapies changed the life of the patients affected by Duchenne muscular dystrophy. I hope that rarer muscular dystrophy like mine will soon have the opportunity to explore this vast field as well. I believe that the involvement of a patient advisory committee in research projects and clinical study is essential. Patients bring a unique and invaluable perspective that goes beyond clinical data. Our lived experience helps shape research in a way that is both meaningful and impactful. By including patients in the process, researchers gain insights into real-world challenges, treatment expectations, and quality of life considerations that might otherwise be overlooked. At the same time, it empowers patients by giving them an active role in shaping the future of research and therapies for their conditions. Collaborations between researchers and patients ultimately leads to a more patient-centered studies, improves adherence and engagement, and ensures that the outcomes being pursued truly address the needs of those living with these diseases. I see it as a mutually beneficial relationship that strengthens both the scientific and human aspects of medical research. Thank you very much.
Thank you very much, Eleonora, and we are very, very happy about your contribution and your participation. And now I will leave the floor to Dr. Orla Lynch, a second patient in our patient advisory board, with a very interesting background, I have to say, as Orla is currently a senior lecturer in criminology and associate dean of graduate studies at University College Cork in Ireland. So her background is in international security studies and applied psychology, but she's also the mother of a 14-year-old boy who has centronuclear myopathy linked to the DNM2 gene. And for this reason, she's very highly involved in research projects and patient networks, being, I'm sure, pretty convinced of the fruitful collaboration of patient perspective in research. So Orla, the floor is yours. Microphone.
That better? Can you hear me now?
Yes.
Yes. Yes, you can. Perfect. Thank you. Thank you for the opportunity to speak. My usual work involves extremism, political violence, and all types of other individuals. So I never thought I'd be bringing my research expertise to bear on something that's so very personal and so very important to me. My son Rian is 14. He's very cheeky. He loves gaming. He's very sarcastic. He wants to be an actor when he grows up, but he has such severe limitation in what he can do physically. And so my motivation to get involved in any of this and all of this work is in the hope that new therapies will become available, but also in the recognition that we need to get involved. We need to give a full picture of what it's like to live with this condition. We need to express the very high cost for individuals and families in terms of their day-to-day exclusion from society, but also the high cost of participating in clinical trials and the expectations around that. So it's a very easy sell when we're trying to convince people of why patients need to be involved in research. We know that it'll improve standardized tests. We know that it'll encourage other patients to get involved. We know that we'll be using the correct measures of success. We know that we'll get good and ongoing feedback. But the morality behind that, to ensure that this is much more than a tick the box exercise, is that the movement called Nothing About Us Without Us a kind of participatory approach to research has been used for centuries at this point in other fields, and it's only now making its appearance into the clinical trials world, and it's a really, really positive thing. And ultimately, it's about including the people that are impacted the most by interventions in the decision-making process. And DREAMS is excellent in that it has really a truly co-development perspective on the inclusion of families and patients. And I think that is the only way forward. I think patients are the experts of their own experience. They need to be included as partners or beneficiaries in large research projects. That expertise needs to be acknowledged, and I think the outcome of doing so will directly impact on a really positive way on the success of trials. So I'm very happy to be involved. I hope I can bring some of my other research experience to bear on that, but most importantly to kind of represent Korean and hopefully be able to move this forward with the input of families. Thank you.
Yeah, I'm sorry, I couldn't activate the mic. So thank you so much, Orla. As you see, it's two very impressive testimonies, and I have to say that our patient advisory board is actually composed of 11 patients, all with their expertise and experience, and really bringing to the DREAMS Consortium a lot of very important perspective. So we have a few minutes for questions, and actually there are already 2 questions which have been asked. So the first one is actually my mistake, but it will make a great transition with the 2 testimonies we just had. I forgot to mention what were the 5 rare diseases which are at the heart of the DREAMS Consortium. So the 5 diseases are Duchenne muscular dystrophy, Emery-Dreifuss muscular dystrophy, centronuclear myopathy linked to mutations in the gene DNM2, Pompe disease, and Danon disease. So for the one who know these different diseases, you can see how different they can be in their clinical development. And so you could ask why we started from these 5 different diseases. And so I will use it to make a transition to the second question, which has been asked, which was, since good AI, so artificial intelligence models, require large amount of data, how do we train artificial intelligence for a specific rare disease that is represented by a small number of patients? So that's a very good question to make this link between the rarity of the disease and the quantity of data needed to train artificial intelligence. So these 5 diseases have at least one molecular mechanism in common, which is called autophagy. And this mechanism is a mechanism which is present in all cells of the body. So that means that there is a lot of research on this mechanism, and artificial intelligence can already learn from all the bibliographic article published on this mechanism to understand what are the different molecules important for this mechanism to function. And then the artificial intelligence can also work on what has been described in each of these 5 pathology in terms of defect or dysfunctionment for this mechanism. So even if for each of these pathology there are few data, we are looking at mechanisms which are common to these 5 pathologies, but also common to the global muscle physiology in general, and even the cell physiology in general. And so artificial intelligence can work on this common mechanism and use all the data available on this common mechanism to try to identify what is altered in this pathology and how to act on this alteration. I do not see other question right now from the audience. So I see, I got a question for Edoardo. So Edoardo, how do you select the prompts you used as outcome measures? And was there patient involvement during the selection procedure for the prompts to be sure that the measure is the most important for the patient? or is it based on frequently reported symptoms? So yeah, Edoardo, if you can comment on this use of prompts and the role of patients in this prompts building, I would say.
Well, this is a very timely question because selecting prompt is very complicated. We need to define a patient-reported outcomes based on the clinical presentation of the disease, of the most frequently reported outcomes, and we need to select based on the ability to measure a change. For example, for the adult treatment of SMA patients with disease-modifying therapies, we were facing a lack of scale able to detect change after the therapies. And we decided for studies, clinical studies, for example, to adopt registration, for example, of the voice in order to detect if there was an amelioration of the dysarthria, to measure if there was a better strength of the voice. And also, for example, we decided to quantify how the patient could perform movements that was not able to perform before, for example. Of course, these need to be tailored, and I think the new novel technology will help us to measure and detect change that we are not able to detect with common scale. But it's not an easy task, and we need to compare what has been described as symptoms of the disease, what we can report as timely and consistent, and how to measure it with the help, in some cases, of different technologies.
Thank you very much. So we got another question. Did you consider the regulatory bottlenecks when thinking to apply for market authorization? So yeah, that's clearly something we integrated in the process right from the beginning, and not even for the market authorization, but even for the clinical trial approval. So the DREAMS project is funded for 5 years. And so there will be a lot of researcher teams involved to try to identify these drugs. And hopefully we'll have drug to test at the end of these 5 years. But we have 5 years to start discussing with EMA and all the regulatory agency on the approval of the protocol of the clinical trial. And so we already plan to meet with some of the task force of EMA, which are dedicated to innovation in clinical trials at month 18 of the program, and then to have a regular discussion with these committees during the last 2 years of the consortium, so that when the 5 years of the program are done, and hopefully we have a drug and we have a protocol ready to start, we can submit this protocol to regulatory agency and have it accepted to test the drug on several diseases. And if the clinical trial is well done and if the drug is efficient, hopefully it will also lead to an easier market authorization approval. So it's already fully integrated in the work program of DREAMS. I'm checking if I missed any question. So we got a question again on the prompts. Often the patients are very heterogeneous, which make it even more difficult to select a single prompt. And for example, the example of the walking test, of course, cannot be applied to people in wheelchair. And so how do we select the correct prompt in this case? Again, will Edoardo or Gérald wants to answer, or I can?
Yes, yeah, in the case we have a patient with different severity of disease, different functional ability, I think we need to select a shortlist a group of prompts that could like cover the abilities of the different group. This is the only way. But again, we need some kind of standardization in order to be sure that these are sensitive enough to capture a difference, and also to be reliable, because of course, in every clinical trial there is a placebo effect, and there is also a different insight, a different way of perceiving the change that's happening when we are ongoing a clinical trial or a treatment.
Gérald, a comment? Your mic is still on.
Okay, no, I do agree, but if we want to have a clinical trial that could be performed in a reasonable time frame, it could be necessary, as I mentioned, to involve to focus on a specific population, and could be independently on the age, and what we can consider is could be the profile of disease trajectories independently on the age, but the homogeneity within a clinical trial is unfortunately necessary to be able to show something that could be later applied for other population. Especially when we perform a clinical trial, what we do as well is to prepare a pediatric investigation plan, which include additional studies for other populations, in order to confirm that what we observed with a specific within a specific population in a reasonable, once again, time frame could be applied as well to other populations. But we have to make compromise to have a clinical trial that could be a success and could be achieved in reasonable, I would say, burden for patient and cost and feasibility for the sponsor.
Thank you very much. And so I think we are reaching almost the end of the webinar. So I'll take the last question on basket trial. So is there more information available on basket trial? And how do you select a common biomarker molecular feature to be used in this basket trial? So the basket trial have been used a lot in diseases like cancers and oncology, already used in rare cancer, for example, but it's not widely used, the less to say, in rare diseases. And so that's one of the challenges, to use what has been done in non-rare disease, in common diseases, to apply them to rare diseases. And as you mentioned in your question, one of the challenges is to find a common biomarker or molecular feature to be measured. And that's why we built this clinical expert committee and this patient advisory board. And they are all working to look at these different biomarker, molecular features, PRO, which could be used in the basket trial. And right now, we already identified a few of them which could be used. But one of the conclusions could be not to have just one marker, but a combination of marker, which could be used to measure a set of features which could be common to the different diseases. But that's really the main role of our clinical expert committee and patient advisory board. It's to discuss and work with us to identify these common biomarker, molecular features, outcome measures to be used in the clinical trials. So we are just 5 minutes out of time. I think, I hope the webinar answered most of the questions you had. We really tried to show that DREAMS is trying to tackle very important issues for rare diseases by combining all the experts, clinical expert, but also patient expert. And we really appreciate the fact that there were so many people attending this webinar.